Good day, dear students. In a series of lectures in microbiology, today we are going to discuss and study regarding the structure of bacteria. As you already know by now that bacteria is a prokaryotic organism, having a size which is not visible by naked eyes, and normally a typical representative of bacteria we consider Escherichia coli has a size of approximately 0.5 to 0.75 micrometer in diameter, and the length ranges from 1.5 to 1.75 micrometers. So point is, it is extremely small in size. It is a unicellular, that is the organism with just one cell, and is prokaryotic in nature. Therefore, whatever the functions the cell has to do has to carry out on its own. No cellular differentiation is there, and all components of the cellular system are enclosed in just one cell. All functions, all metabolic activity, the movement, the reproduction, growth, everything has to be performed by a single cell. So this is a very unique thing in a sense that the cell has to be competent enough to perform everything on its own. And this is basically due to its structure. So now let us discuss in detail regarding the structure of bacteria. This is a structure of a typical bacterial cell as shown in this diagram. If we discuss the major components of a bacterial cell, we will start from the outermost layer. The outermost layer, which is shown in this case, in this diagram, as a dotted structures like this, which are at the outermost part of the cell, it is capsule. This layer is just outermost, and be careful, it is not present in each and every cell. Next to it comes the cell wall. This part is cell wall. The whole of this is a cell wall. The cell wall will vary. We will see in detail the structure of each and every components. After cell wall, what we have is a cell membrane, cytoplasmic membrane. Many times it is also known as a protoplasmic membrane. So cell membrane or cytoplasmic membrane comes beneath the cell wall. So these are the external organs. But in these external organs, apart from capsules and the cell membrane, we have two important things, that is flagella and pili. They have their own functions, but they are located outside the cell. Then comes what are the constituents present inside the cell. Then what we have is mesosome, granules or inclusion bodies, we call it, these kinds of things, granules. Here it is shown, the inclusion bodies which are shown here. Then we have a granular structures which are known as ribosomes, the site of protein synthesis, and the most important component which is a hereditary material and controls all the functions of the body, is this thread-like structure which is spread over the entire cell, which is distributed over the entire cell, is called nuclear material or nuclear body. Remember, in case of prokaryotes, it is not called nucleus. However, in a routine language, few people or few books may mention it as the nucleus of the bacterial cell. But what they mean to say is actually not a nucleus, but it is a prokaryotic nuclear material. So it can be addressed as nuclear material, or it can also be addressed as nucleoid. So this is about the structures. There are other things which are also present in some specialized bacteria, but we are at present studying a typical representative of bacteria, and that is E. coli. There are two figures. This one is rather more clear. That is why I have kept two figures at the same time. As described earlier, as just discussed earlier, we will classify into two categories: the structure outside the cell wall and structure inside the cell wall. As I have already told you, the structure which are outside the cell walls are capsule, flagella and pili, and the cell wall itself. But in few specialized organisms, we also find the other structures like sheath, prosthecia and stalks. This we will study in detail when we study about the morphology of bacteria as a separate lecture. Now, when we talk about the structures inside the cell wall, naturally, cell wall can be considered as the outer layer or it is the inner layer. But then we have cytoplasmic membrane, nucleoid, mesosomes, ribosomes, the cytoplasm itself, the living fluid which is contained in a membrane, the cytoplasm itself. It is a gel-like structure. And one of the specialized structure which is unique in bacteria, not found in all bacteria, but in few bacteria like bacilli, Clostridium, Sporolactobacillus, is a spore. These spores are often spoken as endospore. Endo means inside. Since they are formed inside the cell, they are known as endospores. So now it must be clear which are the structures which are outside the cell wall and structures inside the cell. Now let us first of all discuss capsule. One by one we are going to discuss all these structures in detail, and the first one is capsule. Now capsule is unique in a sense that it is a very, very viscous, slimy. Slimy means sticky layer. Because, why it is sticky? Because it is made up of polysaccharides like dextrans and levans. These are the polymers of simple carbohydrate, monomeric carbohydrates like glucose. So dextrans and levans, like they are polysaccharides. And this makes the capsule slimy or sticky or gummy. These are the different words which are usually mentioned along with the characteristics of a capsule. The thickness of this capsule is very, very thin, approximately 0.2 micrometers in thickness. In few cases, where certain bacteria produces a great amount of capsules, like say for example Xanthomonas, this size can be up to 0.5 micrometer. Most of the capsules, as I have told you, is made up of polysaccharides, but few of them can be made from peptides. that is, it is a proteinic in nature, like in Bacillus anthracis, which causes the disease anthrax in sheep, in cattle. This capsule is made up of peptides. But if you see the overall composition, then mostly in 95% of the cases, capsule is made up of polysaccharides. Since it is slimy, it is gummy, about 98% of the dry weight of capsule is water, and the remaining 2% is either polysaccharides or peptides. Now, these capsules, which are found on bacteria, not all, in few species, they can be categorized into two different types. First one, the capsule itself, and the second word, which is commonly used in many cases, is slime layer. What is the difference between the two? The difference between the two is capsules are firmly attached to the cells. They are firmly attached. Firmly attached means it is very difficult to remove capsules. However, in case of slime layer, these capsules can easily be lost even when we prepare a smear and put the slide underwater. It means they are loosely attached. So it is a difference of attachment. And since it is loosely attached, that we call it as a slime layer, the shape of the slime layer is more or less irregular because there is no firm attachment. While in case of capsule, capsule is firmly attached to the cell wall, and hence whatever the shape of the cell is there, the capsule will be of that shape. It may be spherical in case of cocci, while in case of bacilli, that is the rod shape, it can be rod. Now, what are the functions of this capsule? What is the importance of capsulated bacteria? The primary importance of the capsule is it helps to prevent desiccation and drying. What do you mean by desiccation and drying? Wherever the availability of water is less, we call it as a dry area or a dry conditions. So, the presence of capsule prevents the cells from desiccation or dry. Secondly, because it is sticky, because it is gummy, it helps in attachment. Now, in what way this attachment will help the cells? It will help the cells to attach to some firm surface so that it is not washed away. Say, for example, to certain solid materials. It also helps to attach among each other and form a slimy structure that is known as a biofilms. Such biofilms are very common, even ranging from our teeth to the various environmental locales, various environmental niches. So the presence of biofilm is a new science which is being explored in detail. So that is the first one, attachment, prevention of drying, and the third one is in many bacteria it is one of the virulence factor. It means it imparts protection against phagocytosis by WBC. Say, for example, whenever an organism enters into our system, specifically into tissues and into our body, blood streams, primary line of defense is skin, other things, skin, hair and other things, mucilaginous layers of the body. But the secondary defense system includes phagocytosis. WBCs, they phagocytize, macrophages, they phagocytize the cells because it is one of the defense mechanisms provided by nature. And therefore, non-capsulated bacteria are easily phagocytized, while those bacteria which possesses capsules, they are prevented from engulfment by WBCs and macrophages. In this way, it can escape the phagocytosis and as a result can cause a disease in an individual. Examples are very many, but one of the most prominent example is Streptococcus pneumoniae. So this is about the functions of capsules. The slime layer, as I had already discussed in detail, these two structures will clearly help you to understand the difference between capsule and a slime layer. See, slime layer is loosely attached here. This is a cell and this outer layer is cell wall. And this slime layer is loosely attached. It is in no way direct contact with the cell. It is loosely attached and hence the shape is also irregular. While in case of capsule, this is the one. And see here, it is firmly attached with the bacterial cell wall. And as I have told you earlier that these capsules also helps in formation of biofilms, that is the function of the capsule in attachment, adherence. You can call it as an attachment to some solid surface like say plastic, wood or even leaves or twigs, whatever is there in the environment. As I have told you, they form films on our teeth also. Capsulated bacteria, they form a biofilm on our teeth, and that is why we have to regularly brush our teeth. Here in this diagram, you can see formation of a biofilm. This is the biofilm in which the cells are there, not one, many, and this is a plastic surface. Now, before switching over to the next structure, I would like to give you one important word, that is exopolysaccharides. Exopolysaccharide is also another name for capsules and slime layer. The name itself indicates exo. Exo means external, outside. And what is the chemical nature of this capsule or slime layer? It is a polysaccharide. And therefore, it is often referred to as exopolysaccharide. Now, the next structure is bacterial flagella. Flagella are hair-like helical structure and they are embedded in a cell wall. It means they arise from the cell wall. These are hair-like structures. And these structures have a uniform diameter throughout their length. In case of eukaryotes, or for example, in case of certain fungi, similar structures they exist, but they are like a whip. Whip means chabuk. But whip is thicker from the place where we hold it, and it gets thinner as it distances itself. So thicker at one side and it will thinner out at the other end. But in case of flagella, the structure is having a uniform diameter. And these structures, they are extremely delicate. Extremely. Why I am saying extremely delicate? Because even during handling of these cultures, the violent forces in suspension or when we prepare the smear on the slide, these structures can be lost easily. Why? Because of their very, very thin diameter. The diameter of flagella is just 15 to 20 nanometers. However, the length can be many times the size of bacteria. The length can be up to 5 times the size of the bacteria. That is, it can be up to 5 micrometer or 10 micrometers also. So, point is, they are helical structures. Why they are known as a helical structures? Because the constituent of flagella, the flagella is made from a protein which is known as a flagellin, and this flagellin protein arranges itself in a helical fashion. It is synthesized in the cytoplasm, but then this structure is transported across the cell wall and membrane, across the membrane and then in cell wall, where it keeps on growing, where it is accumulated and it grows there. So this protein, flagellar protein, is also known as H, capital H, H protein. H stands for Houch. And what is Houch? It is something, because the growth of these flagellated organisms on a plate is a spreading growth, because the function of the flagella is locomotion. For a movement of the cell, it imparts locomotory movement. Flagella can move, cell can move with the help of flagella. It has a rotary motion, as we will see it in structure. And when it grows on a plate, it forms a spreading growth. And that is why it is compared as a Houch, H-A-U-C-H. So, many times it is known as H antigen. Okay. These flagella are attached on the cell in a different manners. And on the basis of them, the cells are categorized into, they are classified into monotrichous, lophotrichous, third one is amphitrichous, and fourth one is peritrichous. Monotrichous, mono means one. One flagella usually situated at one pole of the cell. So when we consider a cell as a certain things like elongated cell, then this is one pole and this is the another pole. So flagella is normally attached at one of the poles of this, where single flagella is there. Normally you will not find the flagella attached here. This is not there. It is mostly either at this pole or at this pole. So it is a monotrichous flagella. In case of lophotrichous flagella, these flagella are attached at one of the poles, but the number of flagella is more than one. As you can see it in this diagram, the red colored bacteria has at least 4 or 5 different types of flagella at one pole. While in case of amphitrichous flagella, as you can see here, amphitrichous flagella, they are found at both the poles of the cell. In this structure, you will find a spiral bacteria where at both the poles, both the ends of the cell, you will find flagella. It can be one or it can be more than one. So they are known as amphitrichous. And finally, peritrichous. Peritrichate flagella or peritrichate organisms are those organisms where the flagella is attached not only at poles but throughout their cell surface of the cells. They are attached at entire surface of the cells. They are known as a peritrichate bacteria. Okay. Now, if you see the structure of flagella, then the structure of flagella is like this. This is, the entire structure is divided into three sections. First one is filament, second one is hook, and the third one is this structure, that is the basal body. Basal body, this is embedded in a cell wall. Hook is a portion which connects basal body and flagella. It is slightly having a crescent shape, that is slightly curved, as you can see here. It is not straight like this, but slightly curved in this way. And at the end you will find a filament, that is flagellin protein. Now this basal body is embedded in cell wall as well as little portion in a cell membrane. Now this is a basal body, consists of several rings, and in the center of the rings you will find a, what we call it as a rod like material, and this is a motor. It is exactly like a motor which rotates, as a result of which the shaft, this is known as a shaft. It imparts the movement to the shaft, and anything which is attached to the shaft will also move. Suppose if we have some belt attached to the shaft, and at the end of the shaft if there is a fan, then because of the movement of the motor, the shaft will rotate and it will impart the rotation to the fan. In this way, what happens is, when a basal body which is embedded in cell wall and cell membrane, the cell membrane imparts energy. How it imparts, that is a different mechanism. There are different things which we are not going to discuss at present, but it imparts energy, and this energy propels the shaft. It rotates the shaft. As a result, this filament, a part of the flagella, will also move. It will also propel. It will move in either clockwise or anti-clockwise direction. And as a result, it imparts locomotion to the cell. The cell will move in one particular direction. Now, the movement of the cells can be attributed to various things. For example, photosynthetic bacteria, they move towards the area where the light is in a greater proportion, and this movement is known as a phototactic movement. Similarly, if the bacteria moves towards some chemicals, the chemicals which are or which can be used for its nutrition, then bacteria will be attracted to that, because the chemical diffuses out and the bacteria will sense that this is something which I can use it for a nutrition. So as a result, with the help of the propellant, this flagella, it will move towards in that direction. That is known as a chemotactic movement. So in short, movement in response to certain stimuli is known as a tactic movement. So what helps in tactic movement, or what makes the cell to move, which organ? It is a. So flagella is one of the most important locomotory organ. Helps in bacteria to move either towards one particular chemical or a light, or it can move away from it if it finds the chemical is poisonous. Or certain bacteria, they do not require light. They want to grow in anaerobic condition at the bottom of the system. Then they will die at the bottom. This movement is controlled by flagella. One thing I would like to that, there are other types of movements which are also exhibited by bacteria. Non-flagellated bacteria can also move, but there are different ways for that. So this was about structure of flagella. Now something similar to flagella is pili. Why I am saying similar? Because it is also a structure which is engraved or attached. It is an external organ which is attached from the cell wall of the cell. But most important thing is this structure is a short needle-like structure. It is a short needle-like structure. Short and straight. In case of flagella, it was long, it was thin, long, and filamentous, helical structure. But pili is a short needle-like structure. And what is the function of pili? Pili helps in attachment. Pili helps in imparting certain antigenic property to the cells. And it also helps the cell in a kind of a sexual reproduction, not exactly sexual reproduction, a kind of a sexual reproduction, you can say what we call it as a conjugation. Remember, this pili, they are straight needle-like structure and they are made from a protein which are known as a pilin protein. In case of flagella, the protein was known as a flagellin. Here it is a pilin protein which makes bacterial pili. Functions, as I have told you, is attachment, and another important function is conjugation. What is conjugation? It is a kind of a sexual reproduction because it helps in attachment with the another bacteria. It can pass its genetic material to the recipient bacteria. Those bacteria which possesses the pili, and these pili are known as a F pili because it plays an important role in fertility of the during conjugation. Exchange of genetic material. Genetic material will be transferred through this pili or a conjugation tube from F plus cell to F minus cells. That is, the F minus means those cells which do not possess pili, and this is known as a F pili. Okay, so this is a function which is very important in case of gram-negative organisms. It is also, pili is also site, or it contains the receptor for phage. So certain phages, they attach on either flagella or on pili, and then through that only they will be able to enter into the cells. Okay, so this was about flagella and pili in short. You can see the difference here. These are black colored which are shown here. They are flagella. They are long. Okay. Sorry, this one. This is a long flagella. And these are pili, needle-like structures. What are these structures then? Certain structures which are similar to pili, they are known as a fimbriae. They are known as fimbriae. They are slightly I would say, or in some cases they are exactly the same as pili. But in few cases they are larger than them. But definitely they are very, very smaller than this structure, that is flagella. As I have told you, flagella can be 5 times the size of the bacterial cell, while this pili or fimbriae, they are very short. So these are the cell surface appendages. Why we call it as an appendage? Because if this structure is lost, it does not kill the cell. They are not very much vital. No doubt, if they are present, they will definitely benefit the cell in some way or the other. But if they are missed out, or if they are removed, if they are lost, cell can still survive. And that is why we call it as appendages, and which are situated outside the cells. Now comes a very, very important structure, that is a bacterial cell wall. This structure has one most important function, is it gives shape to the organisms. The shape of the cell is determined by the cell wall and its composition. The cell wall of bacteria, they are of two types. And on the basis of that, the entire bacterial population, all species, can be differentiated into two groups. One is known as a gram-positive group or gram-positive bacteria, and the other one is known as a gram-negative bacteria. This classification was done by Christian Gram, who discovered a staining technique which is known as a Gram's staining technique. And this Gram staining technique is primarily based on physico-chemical nature of cell wall. So depending upon the chemical and physical nature of the cell wall, a particular stain will be taken up while a particular stain will be washed out during the staining procedure. And hence, at the end of the staining, when we observe under microscope, few bacteria, which we call them as a gram-positive bacteria, appears purple or violet in color, while gram-negative bacteria, they appear pink if saffron is used as a counter step or a secondary step. We are going to discuss that Gram staining, one of the most important staining in microbiology is Gram staining. Whenever we want to analyze any sample, primary thing that we do is prepare a smear, stain it with the Gram staining technique and observe under microscope. And as a result, we will be able to classify the bacteria into two groups, gram-positive and gram-negative. This word, gram-positive and gram-negative word, will be used in future lectures when we study different diseases in detail. Now, as I have told you, depending upon the physico-chemical nature of the cell wall, cells are divided into two categories, gram-positive and gram-negative. What is the difference? What is the difference between the chemical nature of the two different types of cell wall? Then we can say that gram-positive bacteria have a cell wall which is made up of peptidoglycan as a major constituent and certain amount of lipid and teichoic acid. While in case of gram-negative bacteria, the cell wall is made up of peptidoglycan and an outer membrane. The outer membrane is composed of lipid and polysaccharide. Lipid and polysaccharide. In few cases, certain proteins may be involved. Now see the entire scenario. Peptidoglycan is present in gram-positive as well as in gram-negative also. But if we see the structure as such, see here, this is the difference which is shown in between gram-positive and gram-negative cell walls. Here, in case of gram-positive, I told you it is made up of peptidoglycan, but the peptidoglycan is thicker in size. Thick peptidoglycan layer is there. Lipopolysaccharide is absent. lipopolysaccharide or what we call it as a outer membrane, it is absent. And little bit of what we call teichoic acids. And these teichoic acids, they are attached with this peptidoglycan. This layer which is shown here is a cytoplasmic membrane, while this green layer is peptidoglycan. Now compare the figure. Peptidoglycan is thicker in gram positive while it is thinner in gram negative. And another difference, peptidoglycan being the only layer in gram positive, it is the exterior layer, it is the outermost layer, while in case of gram negative organisms, peptidoglycan makes a second layer, inner layer, while the outer layer is this lipopolysaccharide. It is in short known as a LPS, this one, lipopolysaccharide. So in case of gram negative, lipopolysaccharide is external layer outside, and inside it has got peptidoglycan. But this peptidoglycan is thinner one as compared to gram positive bacteria. So I hope this will be clear. Now, the peptidoglycan which is there, it is present in gram positive and negative both. But in case of gram negative, it is thinner, size is thin. So this peptidoglycan, basically, it is, if you see the structure, then it is a glycan backbone. Glycan backbone means glycan means sugars, sugar backbone. And this sugar backbone is made from the alternating units of N-acetyl glucosamine and N-acetyl muramic acid. Two things, N-acetyl glucosamine and N-acetyl muramic acid. In short, it is written as N-acetyl glucosamine and N-acetyl muramic. These two alternating backbone, which is formed by N-acetyl glucosamine and N-acetyl muramic acid, it is linked, since they are there, they are linked by, cross-linked by glycan bonds. And there is a peptide linkage. Peptide 5 amino acids are linked, as you can see it in this figure, this one. This is N-acetyl glucosamine, this is N-acetyl muramic acid, and the linkage here is glycan. While this here, these are the 5 amino acids. Now this is one unit. Similarly, there will be another unit here, and this unit, these repeating units of N-acetyl glucosamine and N-acetyl muramic acid having peptide chain, they are linked with one another by what we call it as a CONH linkage, that is peptide link. As you can see it in this diagram, that this is N-acetyl glucosamine, this is N-acetyl muramic acid, and these round things, they are the 5 amino acids, tail of 5 amino acids. So this will be linked with the another one, this one and this one. This has got a link here, see. So this forms a network which is very strong as well as elastic. So peptidoglycan is a structure which is imparting the rigidity to the cell. Now another one, that is teichoic acid. Teichoic acid is attached to, it is only present in gram positive bacteria, and what is it made up of? It is a water soluble polymer of glycerol or ribitol. Water soluble polymer of glycerol or ribitol. And it is constituting about 30 to 50% of the dry weight of the cell. Means It is a substantial part of the cell wall of gram-positive bacteria. And the function of this teichoic acid is it imparts antigenicity to the cell. It is a very important constituent of antigenicity of the gram-positive cells. Peptidoglycan does not impart much antigenicity. It is the presence of teichoic acid in gram-positive cell wall that imparts the antigenicity to the cell. Okay. Now let us come to the cell wall of gram-negative bacteria. The cell wall of gram-negative bacteria is made up of lipopolysaccharide, and it is nowadays mentioned as an outer membrane of bacteria. Why it is known as the outer membrane? Because the structure of this outer membrane resembles to the structure which is there in the actual cytoplasmic membrane. So cytoplasmic membrane is one of the layer which protects cytoplasm or which covers the cytoplasm, and on the cell membrane there is a layer. In case of gram-negative bacteria we call it lipopolysaccharide or a outer membrane. So what is it made up of? It is made up of three things. One is lipid bilayer, protein and lipopolysaccharide. Lipid bilayer, protein and lipopolysaccharide. Lipopolysaccharide is made up of lipid A and a polysaccharide. And lipid A, it is a phosphorylated glucosamine disaccharide. Phosphorylated glucosamine disaccharide. And remember, it is a strong antigenic in nature. So when we talk about polysaccharide as a whole, there are two sections. One is core polysaccharide and the other one is polysaccharide, means outer polysaccharide. Core polysaccharide structures more or less remains constant, but the outer sugars, that is O layer, O polysaccharides, they change in different species or even within the species also. So this is the entire structure of what we call it as a gram-negative cell wall. Remember, this is the structure which is inner membrane. But why it has been represented here? Because this structure is very much similar to the outer membrane, and that is why that is outer cell wall is known as a outer membrane. See, it is a lipid bilayer. This is how we represent the lipid bilayer structure. One end hydrophobic and other end hydrophilic. So this structure and these structures, they are very much similar. See here. And this is lipid A, the round thing. Here, this all entire thing. And on this we have polysaccharide, sugars, out of which the one which are attached to lipid A, this much amount, it is a core body, core sugars, and this one is the outer polysaccharide. So outer polysaccharides keeps on changing depending upon the species and depending upon the within the species also. This is something which is shown in depth. This is a cell membrane. This is peptidoglycan. In gram positivity the outermost, in gram negativity is inner one. And this is what we call it as the outer membrane. And outer membrane is made up of a bilayer structure, bilayer lipid, and this is polysaccharide. Polysaccharide is made up of lipid A, then core polysaccharide and outer polysaccharide O. So this is about bacterial cell. So now we have dealt with the structures which are outside the bacteria, outside the cell wall, cell structures external to the cell wall and cell wall itself. Now coming into the cytoplasm. One of the structure which is there in cytoplasm. What is there? What is a cytoplasm? That we are going to discuss later. But the most important structure of the cell is nucleolus. What we call it is nucleoid, nuclear material. Bacteria being prokaryote, the nuclear material is not enclosed in nuclear membrane, unlike eukaryotes. And that is why they are known as the prokaryotes, primitive cells. Remember the previous lectures. Their membrane is absent. Nuclear membrane is absent. In case of eukaryote, nuclear membrane is present, and therefore that nuclear membrane encases the nuclear material. And that is why a definite place or a definite dot-like structure is observed when we stain the cells with certain basophilic stains. While in case of bacteria, if you use basophilic stains, then entire cytoplasm is stained like that. You will not find any demarcation. Why? Because in case of bacteria, nuclear membrane is absent, so the nucleus as well as cytoplasmic RNA, both are highly acidic in nature. They will bind with the basophilic dyes and the entire cytoplasm will be stained. So the point is that prokaryotic bacteria, they do not have nuclear membrane, and therefore the nuclear material is the word which we use, or another word is nucleoid. Okay, what is the function of nucleus? Well, it will take many lectures to explain you exactly the function of nucleus, but in a nutshell, it is the genetic material of the cells. The nature is DNA, deoxyribonucleic acid. And you know the structure of DNA as it was discovered by Watson, Crick and Wilkins. DNA is a genetic material. DNA makes the bacterial nucleus, bacterial chromosome. Many times we use the word chromosome. It is entirely different from the eukaryotic chromosome, while the bacterial chromosome is haploid and it is a long, in most cases circular, or in few cases linear structure. But it is made up of DNA, deoxyribonucleic acid, which is made up of a base, adenine, guanine, cytosine, thymine, sugar, which is ribose sugar, deoxygenated, and phosphate. Three things makes the DNA. Now this DNA, it controls all the functions of the cell. Cell is itself a single cell. All functions which are there, but functions to be performed at what time, at what juncture, whether to synthesize the protein or not, whether to synthesize the enzyme or not, how to control the metabolic regulation of the cell, everything is stored as an information in the DNA. And it will be passed on to the subsequent generations, because exactly like the parent, because the replication is asexual. Now, similar to the what we call it is a bacterial nucleoid or bacterial nuclear material, there is some structure which is many times called as a plasmid. The structure called as a plasmid. It is an extra chromosomal self-replicating DNA. Extra chromosomal. Be careful, it is not a part of bacterial DNA. Extra chromosomal. Outside the chromosome. Self-replicating. It can multiply on its own. It can replicate like DNA itself with the same mechanism. But these plasmids are not the part of every cell. Only few lucky cells few lucky ones, they have these plasmids with them. And plasmids can be disposed of. It means cells can be cured of plasmids. Why we use word? Because plasmid is considered as a kind of infection to the cell. If it is removed or if it is cured, then cell can continue function. Then what is the need of plasmid? Bacterial evolution has been kind enough on certain bacteria that it has imparted certain genes, because plasmids are made up of DNA and they can self-replicate itself, and this DNA would carry certain genes, and these certain genes would definitely help the cells for its comfortable and survival techniques. Like the presence of plasmid would help the cell to catabolize certain compounds which otherwise normally the cell would not do it. The presence of plasmid would help in formation of a fimbric, pili, we call it as a F pili, which helps in conjugation. So plasmids can be disposed, or it can be gotten away with it. Cell can function without plasmid, but cell cannot live or cannot multiply without DNA or RNA. So this is about the nuclear material. And be careful, again I am repeating, the nuclear material is spreaded entirely in the bacterial cells for only reason that it is not enclosed in nuclear membrane. Prokaryotes, it is an important feature. There are no membrane bound structures. Another important thing about nuclear material is, in case of bacterial DNA or a bacterial chromosome, it is usually single, that is a haploid, and circular or linear. It remains in a super helical structures, and this is done with the help of histone-like proteins. Histone-like proteins, not histones. Histones are the unique property of eukaryotes. In bacteria, histones are absent. So, certain proteins, you will study those proteins in detail if you happen to study the bacterial replication, DNA replication. But these structures, these proteins, they do perform functions similar to eukaryotic histones. That is, it helps the bacterial DNA to exist as highly compact supercoiled structures. Highly compact means they have to be reduced in size. Supercoiled structures. Otherwise, if you see the size of the bacterial DNA, if we stretch the entire bacterial DNA in a linear form, the size of the bacterial DNA would be more than 500 to 1000 times that of the cell size. That is, it can be around 500 micrometer in length or 1000 micrometer in length, depending upon the chromosome. So, if you stretch from one end to another, it will be 500 to almost 1000 times. But how that long, what do you think that long structure remains in a cell which is hardly 1 to 2 micrometers in diameter or in length? It is because these proteins, they help the bacterial chromosome to wind in a very compact form and make it as a supercoiled structure. Naturally, this supercoiled structure has to be unwounded during replication. So this is about the bacterial DNA. Now comes bacterial ribosomes. Ribosomes, they are the granular material which are spread over the entire cytoplasm. And these are the site of protein biosynthesis. Site of protein biosynthesis. When I say the site of protein biosynthesis, means protein biosynthesis takes place on ribosomes. 3 different types of RNAs, mRNA, rRNA and tRNA. All RNAs, they help in synthesis of proteins. They have different role in the synthesis of protein. So, in just one line, you can say ribosomes, they perform protein synthesis. What is their function? Protein biosynthesis. The same function is there in eukaryotes. But the difference between the 2 types of ribosomes among prokaryote and eukaryote is, in case of prokaryotes, ribosomes are of 70S type, while in case of eukaryotes, ribosomes are of 80S type. Now what does this S stands for? S stands for, I'm talking about S, English alphabet. S stands for Svedberg's unit. And this is something related to the centrifugation process. In case of prokaryotes, there are 70S types, while in case of eukaryotes, it is 80S type. Now, this figure shows you the structure of ribosome. See here, this is the entire ribosome, complete ribosome. But these ribosomes can be differentiated into 2 components. First, larger unit, it is known as, this was 70S, while this one is 50S and 30S. Remember, you don't have to add. Addition is not to be done. Otherwise, 50 plus 30 should make it 80S. But instead, because the centrifugation process not only considers the size or the weight, but it also considers the morphology in which this, the shape of the cells and everything, shape of the structure which you are trying to centrifuge. And that is why the total does not make 80S, it is a 70S. In case of eukaryotes, it will be 80S. Here, in case of prokaryote, 70S, having 2 units, smaller subunit of 30S, larger subunit of 50S. Now, this larger subunit, which is of 50S, what is it? Ribosomes are made up of? Ribosomes are made up of protein and RNA. So, which different types of proteins are there? There are these 2 types of RNA and 31 different types of protein. Remember, 31 different types of proteins plus 2 different types of RNA. One is 23S RNA and the other one is 5S RNA. These RNA which are present in ribosomes are known as the rRNA, ribosomal RNA, ribosomal RNA. And that is why it is known as the rRNA. So, this one, 23S rRNA, 2S rRNA and 31 different types of proteins. While here, in the lower subunit, they are made up of 16S rRNA and 21 different types of protein. 16S rRNA. Very important RNA, because the newer system of classification as proposed by Carl Lewis, based on the evolutionary relationship, is entirely based on this rRNA. 16S rRNA. So this RNA plays a very important role in formation of the lower subunit of the ribosome. So next comes, after ribosomes, we have mesosomes. What are mesosomes? These are the structures which are formed by the continuation of the membrane, and they are considered as convoluted membranous structures. Convoluted means they are made like this. As you can see, suppose this is a membrane, the membrane of the cell. So this membrane is convoluted in this way, or you can say internal invagination, not external, not like this, not outside the membrane, not on the outer side of the membrane, but in the inner side. Inner side means on the cytoplasmic side. So these are formed in prokaryotic cell by invagination of a plasma membrane.